All right, well, welcome back. Today we're going to revisit the idea of kind of looking at a single organism and talk about some of the things and challenges that an organism has to do in order to stay alive. So we're going to talk about life and how a living organism with multiple cells primarily stays alive and the things it has to do in order to accomplish that. And we'll start with this idea of homeostasis. And the idea behind homeostasis is it's an internal balance and maintaining an internal balance. So if you have a living organism, the outside of the world, the environment, is constantly changing. But life has to stay within certain boundaries. So for example, and this is just an example, so it's not that homeostasis is only this, it refers to any of these kind of scenarios. But if you were a human, your body temperature is about 37 degrees Celsius, for example, which is the same as about 98 degrees Fahrenheit. Your body temperature, if you get sick or something like that, it can go up a little bit and it can go down a little bit. But if you go too far above or too far below the little bit, you get out of homeostasis. You end up dying. And all organisms have to maintain some sort of internal balance, even though the outside is constantly changing, because life only exists within certain boundaries. Same thing is true with, for example, the amount of water. Your body's composed of a large percentage of water. You can lose some water and you can have a little bit more water, but within certain reasons, if you lose too much water or you gain too much water, both of those result in you as a human not surviving. And that's true of any animal also as well. So in this particular scenario, as we talk about this, this graph here is trying to show you that, for example, one of the things that's important in your blood is how much calcium you have in your blood. Okay, and so if your calcium levels are too low in your blood, there's this negative feedback system in which essentially what happens, in this case it's the parathyroid gland secreting parathyroid hormone. The hormone is designed to target a signal of events in your body to take calcium from your bones and move that into your blood. And if your calcium levels in your blood are too high, the hormone is stopped being released and you shunt it back into your bone. So it's called a negative feedback loop, in which essentially as one event happens, in this case low calcium, your body has a system of responses that are designed to drive it the opposite direction. That's called a negative feedback loop. Okay? If we were to take way back in the beginning of the semester, when we talked about a single-celled organism and we talked about the cell membrane, if you had a single-celled membrane that lived on a rock down by the ocean, and this organism, single cell, needed to get oxygen in, which it does, for example, for cellular respiration. Oxygen is one of those things that can go right through the cell membrane. As the cell survives and produces waste, one of the waste, for example, CO2, to get rid of CO2, that can go right out of the membrane. Okay? Water, in order to get water can also go in and out of the cell. Okay? So if you're an organism or an animal that's just very small, or something that's only one cell, like some of the protozoans we talked about, getting things in and out of a cell, simply a matter of diffusion or osmosis or facilitated diffusion, that kind of thing. But what we want to do now is say, okay, well, what if that cell, and once again, that's why I'm not an art major. This and many other reasons maybe, right? So let's say that's supposed to be now, that's a cell, but it's like at the end of your finger. Okay, so here's your hand, and that cell's at the end of your fingertip now. Now you have a different problem. In a big organism like humans or any large organism, getting oxygen to the cells at the end of your fingers, for example, that becomes a lot more difficult. And so you need to have systems designed to do that. So one of the things we're going to talk about today is the circulatory system. And here's an example here showing you what essentially happens in a circulatory system is here's the cells maybe at the end of your fingertip or in your brain. And what we do instead of diffusing it from the environment, we have these blood vessels that are designed to carry blood to different parts of your body, and then things like oxygen and CO2 and other nutrients can go from the capillaries in this case, or from the blood vessels, to feed these cells, and then the waste products can be put back in and recirculated around. So that's what we're going to talk about today. Here's a picture just showing you what that looks like in an actual capillary. So here's a capillary here, and here's some red blood cells in it. So these are red blood cells, and you notice that the size of the red blood cell. So that's one single cell. So it's pretty small, and that gives you some feeling then of how big these capillaries are, which are also very tiny. So we'll talk about those later on as well. Okay, so when we talk about circulating blood through a system or a network of blood vessels, one of the common sort of ways to do that is to talk about these basic components: an artery, an arteriole, a capillary or capillary bed, a venule, and then a vein. So generally what's going to happen is blood is going to travel from the heart, which we'll talk about later, in an artery. And you notice that the arteries, they tend to be thicker, they have more muscle around them, they're very durable, they're designed to withstand greater pressure. There's more pressure on the artery. Blood travels down the artery, and then the arteries tend to be fairly big. So what happens is the arteries, as they reach your organs, the arteries tend to branch into smaller and smaller arteries, and eventually they branch into smaller artery-like vessels that we call arterioles. The arterioles then branch down to these microscopic sized network of blood vessels that are called capillaries. So these are very thin, and they tend to be very leaky. So what can happen is the blood generally stays in it, but what will happen is some of the fluid, like some of the water and the oxygen and the CO2 and the glucose, those will travel out of these capillaries, and then all of your cells of your body will be in this area. So let's say this is your brain. Blood will travel from your heart in an artery, heading up to your head, into a smaller artery, into an arteriole, and then your neurons, your brain cells, will be sort of bathed in this network of capillaries, and that's where the glucose and all that exchange occurs. Then the fluid continues back in. Some of that gets pushed back in. The waste products get pushed back into that. And then that flows into a venule, which eventually reaches larger blood vessels and goes into a vein, and then that goes back to your heart. So we'll talk about that. One of the things that can happen is your arteries, like I said, have this muscle, this special muscle, smooth muscle, that can constrict and can relax. And inside there is the space. So the space is designed for the blood to flow through. Various things like your diet, certain lifestyle things such as your diet, smoking, other things related to things you might eat, events you might do, drugs you might take. Those can lead to damage to the inside of that. And this can also be related genetically. Some people just have more of a genetic predisposition for this happening. But you'll notice in here, the difference between this one and this one, this has a plaque in it. And so this artery here is sort of closed off. So this plaque is like an injury, and it's kind of like a scab that occurs inside the artery. And as this builds up, what happens is this gets smaller and smaller and smaller. And depending on where this artery goes, if this artery goes to your heart or to your brain or to your kidney, as this gets blocked off, it can shut off the blood supply to those regions and cause severe damage. So one of the things that happens as this closes off, the space gets smaller, and then things like high blood pressure, for example, if your blood pressure goes up, it can squish that down even more and close that off completely, resulting in things like a heart attack or a stroke and so forth. So we'll talk about that. Next thing I want to talk about is the pump itself, the heart. And there are different types of hearts depending on the kind of animal you're talking about. I'm going to stick to the general 4-chambered human heart just to sort of give you a good example of one of the ways a heart can work. So what happens here, the heart in a human heart has 4 chambers to it. So there's 2 atria. So that's the right atrium, and that's the left atrium over there. And so this, even though that's the right side of the picture and the left side of the picture, you have to always think about this as the patient's right and left. And so this heart, this would be a person, for example, standing facing you, and you would be looking at their heart. So you're, to orient this correctly, the person's looking at you, their eyes are up here, for example. Here's their head, which actually should be up higher, but I'm running out of room. So they're looking at you directly like that, and there's their body kind of like that. And so that's their right side. So it's not your right side, it's the patient's right side. So that's the right atrium, and that's the left atrium. This is the right ventricle. This is the left ventricle down here. And what happens is the atria pump blood from the atria through these valves here I'll talk about in a second, and into the ventricles. So the left atrium pumps blood from the left atrium to the left ventricle. The right atrium pumps blood from the right atrium to the right ventricle. And they go through these valves when they do that. And the valves are designed such that they can open and close. So when they're like swinging doors. So as the blood pressure increases in the atrium, as the pressure increases in the atrium, as the atria contract, it swings the doors open and the blood flows in to the right ventricle, and that's also happening at the same time into the left ventricle. When these are filled with blood, the ventricles contract and the blood starts to move back this way, and that closes these atrioventricular valves. They slam shut on both sides, the left and the right. And often they refer to the heart. If you listen to the heart through a stethoscope, they refer to the sound as a lub-dub. So you hear this lub-dub, lub-dub, lub-dub. That's the sound of the heart. And that sound is the closing of the valves. So the first sound you hear, the lub in that sound, is the closing of the atrioventricular valves. So as the blood flows this way, it closes the atrioventricular valves. On the right side, the atrioventricular valve is called the right atrioventricular valve. It's also called the tricuspid valve because there are 3 pieces to that door. On the left side, that is called the left atrioventricular valve or the bicuspid valve because it has 2 valves on it on the right. So 3 on the right, 3 on the right, 2 on the left. So 2 sort of doors or parts to the door on the left atrioventricular valve, which again is a bicuspid valve. Okay? So blood flows in, and as it moves this way, it closes those doors. The doors close. That's the lub sound. And so the blood then, instead of going this way, travels up this way on both the right and the left. So on the left side, it goes up. It goes behind this blood vessel here. This is the pulmonary trunk, which becomes the pulmonary artery, which goes to the lungs. So this is traveling on its way to the lungs. Behind that, so you can see how this comes up and sort of goes behind the pulmonary trunk, this is the aorta. So the left ventricle pumps blood up through here, and it goes through the aorta. And the aorta then, pumping from the left side, comes up, and this goes to the rest of the body. So the blood here goes to the rest of the body. Okay, I'll come back to that in a second. On the right side, the blood comes up, flows this way, and it's going to end up going to the lungs. So what happens as the ventricles contract, they push blood up this way. It goes through the semilunar valves, and then as the right ventricle and the left ventricle finish their contraction and push the blood up, they start to relax. And as they relax, the blood starts to flow back. And what happens there is that's when the semilunar valves, so the semilunar valves, those will close. And you have a right semilunar valve and you have a left semilunar valve. Those close as the blood flows back towards the ventricle, closes that off and prevents the blood from flowing back in. That's the dub sound. So the lub dub is the closing of the valves. Okay, so the first sound, the lub, is the closing of the atrioventricular valves, both of them together, and the dub sound is the closing of the semilunar valves. Okay, and so on the right side, they draw this in blue and red to sort of let you know that this is what they call deoxygenated blood, and the red is oxygenated blood. But in fact, really not, one's not really blue and one's not really red. They're both basically the same color because this is still 80% oxygenated in a normal working heart and person, whereas this is supposed to be 100% oxygenated, and by that I mean that's the most amount of oxygen it can carry. So blood goes from the pulmonary artery, goes to the lungs, picks up the oxygen, and comes back the pulmonary vein and goes to the left atrium. Left atrium contracts, pushes blood to the left ventricle. From the left ventricle, the ventricles contract, they push blood up. It goes up through the aorta. As the ventricle relaxes, the blood starts to flow back, but it's trapped by the semilunar valve once again on the left side, catches the blood and prevents it from flowing back. That gives you the lub dub sound, and eventually as the heart keeps contracting, it pushes blood to the rest of the body, and then eventually that comes back after it goes to the body. Okay, so this will go up, it'll branch a bunch of times into capillaries, it'll turn into venules, and eventually it will return to the vein, and this is the main vein going back to the right atrium, and that is the vena cava or the vena cava. I hear it pronounced both ways, and that returns blood back to the atrium, which then can go back to the right atrium, and therefore then it goes to the right ventricle. At that point, it's the so-called deoxygenated blood, but again, it's 80% still oxygenated or so, and then it goes back to the lungs to the rest of the body. Okay, so that's the basic flow of blood through the heart, and that's definitely one thing you want to focus on. If you were to measure blood pressure, the idea is to get blood flowing to your tissues, and there are a couple ways to do that. One is you can increase the pressure and increase the flow that way, and you can also increase the rate at which your heart is pumping. So when you take your blood pressure, what you do is you blow up this device here and you listen, or the device is automated, and essentially what you do is you blow this up and you cut off the blood pressure to your artery. And the first sound you hear, or the first beat, is when the blood is at high enough pressure that it can push past the cuff you've put on your arm, and that's your systolic pressure. That's called your systolic pressure, and that's supposed to be somewhere around 120. Then at that point you'll hear your heartbeat continually, and eventually there's so little pressure on the cuff that you won't hear the heartbeat anymore, or your sensor, if it's an automated device, won't be able to pick up the heartbeat anymore, and that's your diastolic. And those two readings, your systolic and diastolic, give you your blood pressure. This is your high end of your blood pressure when your heart's basically beating, and this is the pressure that's still on the artery as your heart is relaxing at its lowest point. That's your diastolic pressure. And a normal person's blood pressure should be somewhere around 120 over 80. As you get into, say, 140 over 90, you get what they call pre-hypertension or hypertension, where your blood pressure is too high, and over time that can result in some serious health issues. Normally your heart rate beats at about 72 beats a minute, and at that rate, 72 beats a minute for the normal heart, you can pump all your blood in your body around pretty good in a pretty short period of time. So 72 beats per minute. As if you're a well-trained athlete and you're in really good shape, a lot of people will have their heart rate down in the 60s because their heart is stronger, and every beat they're able to push more blood per heartbeat, so their heart can beat at a slower rate. If you're not in very good shape, or there's some other issue, your heartbeat might be 100 beats per minute, which means it's beating faster because on each beat it's not able to carry as much blood. This is a resting heart rate, so not during exercise. Okay, several things can happen with the heart. As I mentioned, with that plaque, one of the things that can happen is over time your blood vessels can get clogged. Okay, so here's the aorta here, and coming off the aorta, right off the aorta, the first branch coming off the aorta, these little blood vessels here, these are the coronary arteries. The heart itself is a muscle, and as a muscle, it pumps blood throughout the body, but also to the heart. But what happens is those blood vessels going to the heart can get clogged or damaged, and that results in a heart attack, or also called a myocardial infarction. So what happens is blockage in one of these arteries here can lead to no blood. If you think about this being like a freeway or a street, roads flowing, if you cut off the road, you get no flow to that area of the heart, and that muscle ends up dying there. And depending on the extent of that, you can, basically kill the whole heart. And then therefore ruin the pump. The same kind of idea can happen in a stroke. So a stroke actually can result a couple different ways, but essentially that if you have a blood clot or you have some kind of blockage, but instead of the artery going to the heart, it goes to a portion of the brain, you get the same sort of result. You get blockage in some particular area, and that can cut off the blood supply to parts of the brain, causing brain damage in that case, and that's what you get when you have a stroke. Because of the structure of it, there's a lot of fluid that is around here, and these tiny blood vessels are under less pressure, and so one of the things that can happen as the blood pressure if your blood pressure goes too high, in addition to the clot, unlike what typically happens in your heart, these can actually rupture too. So one of the other kinds of ways you can have a stroke is this can actually rupture, and therefore the blood's not flowing to any region anymore because this blood vessel actually blows out. But that can also result in a stroke. But same idea, basically, you're not getting blood flow to the region of the body or of the brain in this case, where the stroke is occurring. The next thing I want to talk about is blood itself. And blood can be broken down into two categories, two parts. The first is what we call the plasma, and that's sort of the liquid portion of the blood, or the non-cellular part. It's the water and proteins and salt and all that kind of stuff in there. Blood is a type of connective tissue, and as a connective tissue, it has not only cells, but it has stuff outside of the cells that make it a tissue. Salt, potassium, chloride, proteins floating around in there, like I said. There's some gases in there and waste. That makes up the plasma, and most of the plasma is water. 90% of the plasma is water. Then there's the cellular elements. The cellular elements include things like the red blood cells, also called erythrocytes, the white blood cells called leukocytes, and platelets. Red blood cells are primarily used for transporting oxygen and CO2 through the body. White blood cells are primarily used for fighting infections. So we'll talk about that. And platelets are primarily used for blood clotting. So if you get a damaged or a cut on your skin. Platelets are what are involved with making that stop bleeding. Showed you this picture, already once, but just to show you it again, what ends up happening is that your blood, as it travels through, the blood cells, the cellular parts, tend to stay in the capillaries unless there's an injury. And what happens is the nutrients flow out. And they flow out of the cell, they travel around, and the nutrients then feed the cells surrounding tissue, and then the waste products of those cells, CO2, other waste products such as maybe ammonia or amino acids after your cells use up the proteins in it, whatever waste products get pushed back in. So there's a flow as it comes from the artery and the blood is pushed through the artery, it tends to flow out and it tends to flow back in. But what it takes with it, it takes the nutrients in and it puts the waste back out. That then goes and gets cleaned up. It goes back to your heart and gets pushed to your liver and your lungs and elsewhere and gets cleaned up. You get rid of the CO2 when you breathe out, for example. The waste products are pushed through your kidney, for example, and as they're pushed through your kidney, they're dumped out as urine and things of that sort. Okay, so this is a pretty good system here of this flow going in and then coming back out. But what happens is you don't get all of the fluid back in. So you typically get, whatever percentage you have coming out this way, you only get a smaller percentage of that going back in. So the blood pressure over here tends to push it out. But as you get near this end, the blood pressure is lower. Okay, so as you move away from the pump, the blood pressure is lower. But there's a pressure also due to the fluid outside here, and that force stays constant. So what ends up happening is as the blood pressure drops on this side, you tend to push fluid out, but you only put about 99% back in, which means about 1% of this fluid is staying out of here. And because of that 1% you're kind of always losing, you need another way to pick that up. And so what happens then is interspersed with the capillaries here, we have this other system called the lymphatic vessels. So the lymphatic vessels, the fluid that goes out of your capillaries, becomes what is called lymph after that. And you've probably heard of lymph nodes before, and that's where this is going. So you'll notice the green inside here sort of intertwined in there with the capillaries. These are the lymphatic capillaries that are closed on one end. So it's not a flow system like you see in the blood vessels. Instead, they're closed. And what happens is that extra 1% of the fluid gets pushed back into these capillaries, and eventually that all flows back into the heart. And I point that out, and you can see that in this picture here, because that then creates a system where as you dump off the blood and you feed these cells and you pick up that 1% of fluid in the lymph, that lymph then will travel back and will get sort of sampled throughout your body through these lymph nodes. So you probably have heard about this before, but sometimes when you're sick, you get swollen lymph nodes maybe in your neck or in your armpit, and you have lymph nodes all over your body. And one of the things that can happen is these are loaded with white blood cells. So at this point, this is why we blend this in here, but the lymphatic system then, which kind of picks up that extra fluid and eventually jumps it back into your heart, into the right atrium. That extra fluid that you're picked up can be sampled for pathogens and things like that. So that then leads to your lymphatic system and part of your immune system. So you have these lymph nodes scattered throughout your body, as I mentioned before. Those are packed with white blood cells. And it turns out anything that's part of the lymphatic system has with it either these lymphatic capillaries picking up this fluid, or they're loaded with white blood cells. So there's a lot of lymphatic structures, your tonsils, your spleen, your thymus gland, bone marrow, for example. All these tend to be very densely packed with these white blood cells. and designed to sort of sample the blood, or the lymph in this case, that's come from your blood to see if there's anything in it that warrants an immune response. So at this point, we can then talk about the immune system, and I'll first talk about the non-specific defense, or often called the innate system. And this is basically what you're born with. And the idea with this is a defense against any kind of pathogen, any kind of problem. It doesn't recognize, for example, tuberculosis versus the common cold. Later on, then we'll talk about the specific immune system, where you can actually designate a different response for a type of cold or a type of flu. So getting to these non-specific defenses, just as an example, your skin is a good system of protection right there. You know, your skin right there covers up the rest of your body, and it's a living organ with all of these new cells being produced all the time. And underneath your skin is all of your soft tissue that could be invaded by pathogens. So having a burn or a very large cut exposes your inside of your body to pathogens. So your skin, for example, does a very good job of just preventing anything from getting inside in the first place. Inside your respiratory tract, for example, there's cilia. That's what you see here. Cilia are these finger-like projections on the cells that are able to sweep off invading pathogens and just get rid of anything that you're breathing in, for the most part, so you can cough it up or swallow it and simply get rid of it. Those are mechanical barriers. There's also chemical barriers. Your body, for example, if you get infected by a virus, so in this case, let's say you have a cell of yours that's infected by a cold virus, this cell's pretty much a goner, because it's got the virus inside and there's very little it can do. But this cell can produce interferons. And interferons are like signal molecules that will travel over and will bind to your healthy cells and signal that there is this virus floating around. And then your healthy cell here can produce these antiviral proteins that can attack this and prevent it from infecting this other cell. Generally, this cell is a goner, like I said. We'll talk about this, but your immune system will wipe this cell out. But this one will survive, and it can pass that information on to other cells. You also have the inflammation response that can occur, and phagocytosis. So if you have a cut, for example, the wound that you have a cut on has maybe bacteria on it. Those bacteria are infecting your skin, and there's signals coming from your cells that they've been injured. Those injured cells release these chemical signals that will cause this expansion in this blood vessel with things like histamine. It'll get larger in size. Your white blood cells will then come out of the capillaries, which they normally don't do, and they will phagocytize or eat up that bacteria. So you notice this blood vessel here on your finger, it's gotten bigger. This makes this warmer, creates like a little miniature fever in this area, and I'll talk about this in a second, but that warm blood tends to often be harmful to the bacteria. You also get swelling in there, and these white blood cells will go in and clean that up, and that's kind of when you get an infection locally and your immune system is able to take care of that. I just mentioned the fever. Fever is, although it makes you feel really crummy, the idea of a fever, the reason your body will elevate its body temperature is if you have a bacterial infection, often the bacteria can only survive the homeostasis of the bacteria is maybe only around 37, 38 degrees. So the idea of a fever, by raising your body temperature to like 39 degrees, is perhaps it will kill the microorganisms. Nowadays, though, with our antibiotics, you got to wonder whether having a fever, is useful. But certainly for our ancestors, and before we had antibiotics, which was not all that long ago, fever was one of those things that could potentially save you from, an infection. Okay. Then the next thing we're talking about is a more specific response. And so this idea of a specific response involves us sort of looking at a couple more details of the immune system. So in this case, we have this pathogen that's invading the body, and it's a bacteria in this case, but it could be a virus or anything really. But any kind of invader has on it these chemical signals, mainly used for it to recognize other sources of hosts and things like that, and those are called antigens. So an antigen is a chemical found on a pathogen that can start your immune system in some sort of response. So an antigen is on the invader, and it has to bind to an antigen receptor of some sort. The antigen receptor is on your cells. So in this case, I'll talk about this in a little bit, but in this case we have a special kind of white blood cell called a B cell. The antigen binds to the antigen receptor, and it can initiate a response. And there's a variety of responses that can occur. One thing that can happen is your body can make antibodies to that. Oops. All right. Can make antibodies. Antibodies are proteins that are secreted specifically by B cells that will attack an antigen. There's a variety of ways they can do that. In this case, they're showing you the antibodies actually binding to the antigen, and then often this thing can be eaten up, or it can be pushed out through your kidneys in your urine. There's a variety of ways actually, but once the antibodies are bound to it, the recovery from the infection is sort of well on its way at that point. There are two types of these lymphocytes. Lymphocytes are really so a lymphocyte is a type of white blood cell or leukocyte. Okay, so a white blood cell, also called a leukocyte, are broken down into different types of leukocytes. Lymphocytes are one of them, and lymphocytes are very important for your specific immune response. And there are two main types of lymphocytes. There's T cells and there's B cells. And I'm going to simplify this a great deal because it's actually much more complex, as I'll sort of hint to in a little bit. But T cells are what we call cell-mediated immunity. T cells come from, first of all, they come from your bone marrow. So all your blood cells actually start being produced from stem cells in your bone marrow. And then those T cells, in the case of T cells, the immature lymphocytes, have to travel to your thymus through the blood, and there they become mature T cells. Your B cells actually stay in the bone marrow and mature there, and they come out as mature B cells. So your T cells function in what's called cell-mediated immunity, and your B cells function in what's called either your humoral immunity or your antibody-mediated immunity. And I'll explain both of those here in a second. I want to point out, this is the thing I was saying, is to kind of show you the complexity of this. So I'm going to show you what B cells can do and what T cells can do is sort of separate. But really you should get in your mind that it's a complex system where they interact. So what happens is you have an antigen. As I mentioned before, an antigen is like a chemical that is on the organism. Okay, so you have this antigen, it's on, let's say, a bacteria that infects your body. That antigen gets picked up in a variety of ways. It can activate B cells directly, or you can have another type of white blood cell called a macrophage or an antigen presenting cell, APC, and the antigen presenting cell can take that antigen, stick it on the surface, and then can activate what's called helper T cells. And helper T cells then can activate B cells or also T cells. Those can then form memory helper T cells, and the B cells can give rise to both plasma cells, which produce antibodies, and memory B cells, that can basically function as a memory of that response occurring. So although I will show you here what B cells do and what T cells do sort of separate, you should realize that in reality there's a lot of interaction that goes on between the two of them. Okay? So once again, this picture here shows you, okay, here's the pathogen, and the pathogen can be all kinds of things. It can be a virus, it can be a bacteria, fungus, parasite of some other sort, or protozoan. You can have self-antigens that are designed to recognize, hey, this is a blood cell that's supposed to be in my body. You can have foreign proteins, for example, and those get engulfed and presented by these antigen-presenting cells that show that to, for example, T cells. And T cells will bind to that and will then be stimulated by the antigen-presenting cell. What happens with B cells directly is if antigens, like let's say a particular bacteria is transported in your body, there's a whole bunch of different B cells you have with different antigen receptors on them. In this case, they're showing you, okay, here's 5 different kinds of B cells. And you notice that this antigen only binds to this specific shape of this antigen receptor, and therefore this B cell gets stimulated into growing and reproducing, and you end up with a whole bunch of those as it goes through mitosis. You end up with a bunch of those. Some of those will go on to form plasma cells that will produce antibodies. And now you have all these antibodies that can then go attack this antigen. Some of these will form memory cells so that if you're exposed to that again, you have a quicker response to that. Okay? So one sort of way to think about this is, well, let me do T cells first and then I'll explain it. Let's say you have one of your own cells. So this is an infected cell. So let's say you get the flu. The flu is a virus, and the virus gets inside your cell, and as viruses do, they can't really reproduce on their own. They have to take over your cell. So when it takes over your cell, what ends up happening is your cell is now infected with this virus. Once your cell is infected with that virus, it's very difficult, if not impossible, to get the virus out of the cell. What happens is your immune system comes along and it will actually kill your own cell infected with that virus using what's called a cytotoxic T cell. It'll bind to it. It'll put this perforin molecule that actually punches a hole in it. It prevents homeostasis then of your own cell, and your cell basically ruptures and it dies. So this is your own cell being killed by one of your own cells. And the idea there is that this cell, because it's infected with a virus, you can't really save it. The only thing you can do is get rid of it and kill it so it doesn't spread the disease. So T cells are really good at killing your own cells that are either infected with a virus, for example, or very similarly have cancer. So if your cell develops cancer and is damaged, one thing that T cells are good at is they can go directly and attack a cancer cell and kill that cell. B cells, on the other hand, because they secrete antibodies, B cells do not have to be directly connected to the cell. The antibodies can travel in the blood and they can attack, say, a bacteria or a virus that's floating around loose in your blood. So T cells are good if you have your cell and the infection's inside your cell. T cells are good at killing that cell with the infection inside. B cells are really good at killing an infection that is, say, in your blood, but it's outside the cell. It's floating around loose inside your blood. B cells are good at attacking that. once again, there's a lot of interaction that goes on between the two. So I'm trying to simplify the difference between a T cell and a B cell, but realize that they're both sort of working together most of the time. In this picture, you can see your primary immune response and your secondary immune response. The idea here is if you get exposed to, say, a type of flu, it takes you a little while to sort of respond to that, and this is your antibody level in your blood if we were to measure that. So if you get exposed to a flu that your immune system has never seen before, you end up getting sick because your immune system has a slow response to it in general, and your antibody level never gets very high. But because of the memory B cells and T cells you produce, if you're exposed to that cold or flu again, what often ends up happening is very quickly those memory cells will produce antibodies very, very quickly. And you notice that here you get this really, really big response to that same colder virus, which is why you tend to only get, many diseases only once, because your body, your immune system adapts to that in the sense of producing these memory cells. So as long as the organism doesn't change very much, you often, for example, for colds, as you get older, you tend to get less colds because your immune system develops a memory to sort of each one. In this picture here, this sort of puts it all together. This is a graph showing you your immunity. Your immune system can be broken down into what you acquire, meaning this is what is you're sort of exposed to over time versus your innate immune system or non-specific. So inborn resistance, like your skin or the cilia in your lungs or the tears that wipe out infections that are in your eyes, things like that, the acidity of your stomach. Those are things that sort of wipe out sort of any pathogen, no matter what it is. But as you get exposed to different forms of infection, viruses, bacteria, so forth, you can develop either active immunity or passive immunity. Passive immunity is acquired, but you get it either by getting across the placenta, for example, before you're born, or in breast milk. So you're getting natural passive immunity, meaning that your immune system is not actively acquiring the ability to fight it off, but instead what is happening is you're getting the antibodies transferred from mom, for example, to fight off an infection. Artificial passive immunity is like when we inject a antibody or an antitoxin. If you got bit by a snake, for example, they could give you antivenom or antitoxin. They inject it, and they're basically injecting antibodies to that. Active immunity is when your immune system has responded to the pathogen. And you can either get that naturally by being exposed, for example. This is what happens with most things. If you're exposed to a cold or virus, you develop naturally acquired immunity. You get sick maybe the first time, you develop memory cells, you're less likely to get that again. But in a lot of places, like in the United States, instead of you just getting sick, we're able to induce active immunity. So what we often do with a vaccine is we'll inject part of the antigen, and your immune system will respond, develop memory cells. So most of the time, if it's done correctly, your immune system acts as if it's sick, but you yourself do not feel sick, or you just mildly get sick, and it prevents you from actually getting the disease.